Hello students, welcome to EPG Pathshala. I am Dr. Vibha from TERI. Today we are going to talk on module Somatic Embryogenesis under the paper Plant Biotechnology and Crop Improvement. In this module, you are going to learn about direct and indirect somatic embryogenesis, factors that affect somatic embryos, induction and development of somatic embryogenesis, physiological and biochemical aspects of somatic embryogenesis, molecular aspects, and application of this important technology. Somatic embryogenesis. It has two words, somatic and embryogenesis. Somatic is somatic cells and embryogenesis is normal formation of embryo. So it essentially means formation of embryos from the somatic cells. So it involves development of somatic embryos from somatic cells irrespective of their ploidy level and their specialization. So essentially it means, and you have already learned about totipotency, that cells are capable to form or differentiate into embryos again. It's a method which is used for mass propagation as well as generation of transgenic plants. And it's also a very important tool for basic studies on cell differentiation, gene expression, and molecular genetics. Now this depicts embryos. So you'll find that from a mass of embryos being regenerated. Way back in 1900, Heberland has proposed that a single vegetative cell could develop into a complete embryo. Now it is that he only, in a way, he speculated that such a thing can happen, although he was not successful in getting embryos. It was thereafter, and it was a wait of 58 years, that when two independent groups, Stewart from United States and Renner from Germany, they succeeded in developing complete carrot plants through somatic embryogenesis. And thereafter, the growth or progress in this area was so fast that today we have more than 500 species which are known to form somatic embryos. There are two ways in which embryos are formed. One is the direct embryogenesis and the other one is indirect embryogenesis. Now, what do you mean by direct? Direct embryogenesis is when the somatic cells get converted into a somatic embryo without any intervening callousing phase. The other possibility is that the differentiated somatic cell first undergoes de-differentiation, that is, it forms callus, and thereafter, on changing the media and other conditions, you induce embryo formation or somatic embryo formation. In case of direct somatic embryogenesis, it is expected that plantlets produced will be mirror images of the mother plants. However, we cannot apply this to chimeras, as in that case, depending on the cell type from which the plant originate, you can get plantlets which are either green or white or whatever the chimeric tissue is like. In case of indirect somatic embryogenesis, explants first produce undifferentiated cells, which then differentiates into somatic embryos. The callus cells may show variability, and so will be the plantlets originating from them. And that is where we also look into variations, that can we induce the kind of variation we want, especially in the vegetatively propagated plants such as banana, where you don't get heterozygous plants. It's always produced through suckers. So if you want to induce traits like resistance to fusarium, which otherwise is not known to occur in that genera, then you can subject this mass first to mutagens and thereafter you regenerate somatic embryos. Before the development of somatic embryos, the plant cells are subjected to biological pattern of redifferentiation, and thus there are many associated factors affecting somatic embryogenesis and development of somatic embryos. Somatic embryogenesis involves a thorough selection of explants, careful selection of nutrient media, plant growth regulators, and the physical environment during incubation in the growth rooms, including light and temperature. The experimentation underlying the development involves series of processes for stimulation of embryogenesis, development of embryogenic cells, and their transformation through germination into complete plants. Usually, shoot tips, immature embryos, and young floral parts have been used as explants. In monocots, that is palms and cereals, and most diocots, the most common explant is immature zygotic embryo. In case of mature embryos, protodermic cells of cotyledon form somatic embryogenesis. The growth regulators provide stimulus for embryogenesis. Auxins occupy the most prominent position regarding tissue regeneration. Different concentrations of synthetic auxins, especially 2,4-D, is used for callus induction. Its concentration varies from 0.5 to 1 milligram per liter, which is good both for callus induction and proliferation. While callus proliferation on auxin supplemented media to induct embryogenesis, it must be transferred to cytokinin-rich media. Of course, various cytokinins have been used, including zeatin, but BAP is the most commonly used. BAP, along with IA, sometimes even use of ABA, gibberellic acid, has proved to be good. For somatic embryogenesis, pH modification and high sucrose concentration without growth regulators have also found a place. Requirement of nitrogen is considered as the second major requirement for induction of embryo formation. It is required both for initiation as well as maturation of embryos. Ammonium chloride, ammonium nitrate, casein hydrolysate, range of amino acids have been used for induction of embryogenesis. The most commonly used carbon source, as for other tissue cultures, is sucrose. Cell density, ethylene concentration, and light are other factors that affect somatic embryogenesis. It's also observed that the initial cell density is the critical factor for differentiation of somatic embryos. In carrot, high density culture on auxin-free media resulted in inhibition of somatic embryo formation. It's because the cells which are cultured at high density have tendency to release high amount of phenolic compounds which inhibit embryo formation. But then there is also a minimum density requirement because embryogenic cells also release promoters. In the next couple of slides, we are going to discuss what are the steps involved in somatic embryogenesis. So as we have discussed, you take the explant. It can either go for direct embryogenesis or it goes through the process of callus formation. And that callus, we can either let it grow on the semi-solid media or we can have suspension cultures so that it grows as a suspension. Now, there are two stages. One is that of embryo induction and the second one is of embryo, further growth of the embryo and its maturation. Now, both have different sets of requirements. That's what we explained you earlier. First set is where you require large number of cells, which is auxin containing media for callus proliferation. In induction, you use cytokinin and that is where it proliferates and a lot of embryos are formed and you actually have a mass with cells showing different or with embryos at different stages of development. Thereafter, in most cases, we transfer this to semi-solid media for further maturation and growth. This slide shows you development stages of in vitro somatic embryogenesis in carrot. Let me tell you, carrot is the most studied system for somatic embryogenesis. It also happens to be the first system in which somatic embryogenesis was successful in terms of complete plantlet formation. So when you start from the carrot cells, you just take carrot and cut it into small pieces and thereafter you make suspension culture. Those starts growing its callus formation which is then converted into suspension. And from that suspension, the embryo starts developing. And this is what it shows, that you have embryos, then the embryos maturate and then they germinate and you get again a complete plant. Research has proved that cells acquire embryogenic competence of physiological and biochemical changes prior to the morphological differentiation of somatic embryos. There are changes in the endogenous level of phytohormones, various metabolites, and so on. So that is what triggers differentiation of embryos. The polyamines, they are also associated with the biosynthesis and again extremely important for somatic embryogenesis. Now you will realize that when embryos are getting formed, they are at different stages of development, and different stages they have different sets of requirements. So one of the steps which is essential for the success of somatic embryogenesis is synchronization of somatic embryos in the induction media, so that when you shift the embryos to maturation media, they all are at the same stage. And this is achieved by different methods. One of the methods is that you restrict the growth at a certain phase. You say that this is where it's G1, G2, whatever stage you want them to be, the cell cycle. So then you collect the embryos which are at the same stage. During the development of somatic embryos, transcriptomics of several species is carried out. Different genes such as LEC, WUS, FUS, auxins and transcriptional factors have a predominant role during the induction of somatic embryos. That is transformation of a somatic cell into an embryogenic cell. So there are a lot of genes which play an important role. Epigenetics controlling the expression of genes has significant role, and research has proved that auxins, in addition to modification of DNA methylation patterns, influence the embryogenic cells. Other factors that regulate proteins are post-translation modification, protein turnover, and protein-protein interactions. As the embryogenic cells move towards embryogenic developments, changes in gene expression occur. Molecular markers indicating this transition have been recognized, and also the genes that enhance embryogenic response have been cloned. The position of the callus and appearance of calcium in the vacuoles are the first signs of recognition of embryogenic cells. Similarly, cell wall associated proteins also participate in embryogenic process. It is well documented that embryogenesis in fresh cultures can be induced by addition of cell-free conditioned media. This is achieved due to the presence of extracellular proteins, what we call ECP, which is released by embryogenic cells that are present in the conditioned media. The zygotic embryos typically undergo a maturation phase during which they acquire tolerance towards desiccation, store food, and sometimes synthesize ABA to induce dormancy. Somatic embryos, however, do not have the stage of embryo maturation and germination, and they germinate immediately, resulting in development of a weak seedling. Thus, morphologically mature embryos of many species require additional stages, something similar to zygotic embryo formation. For example, gradual desiccation, increase in sucrose concentration, and ABA treatment during maturation stage help in better survival of plantlet development from somatic embryos upon transplantation. To improve germination of somatic embryos, reducing humidity inside the culture while mimicking embryo sac environment also favors survival of embryos in vitro. Because ultimately, success of embryogenesis is how many embryos are formed, how many of them you could successfully transfer to the field. And that dictates its application both for research and for commercial applications. Now coming to the practical applications of somatic embryogenesis, you have understood and appreciated the large number of cells that can be produced from a small, very small explant. Now that explant gives rise to callus, which potentially can be multiplied indefinitely. So once you have initiated a culture, that becomes a bank for you to multiply cells anytime. And on subjecting these cells to different conditions, to different set of cultural conditions, you can get formation of embryos. Long back, it was also thought, and to some extent it still holds true, that is that the cells, there is certain threshold. So it is usually the diploid cells which will give rise to somatic embryos. Now, what does this mean? One of the most serious objection against callus is that callus cells divides very fast. It's not all the time that the chromosome duplication, the complete set of chromosome duplicates. It's not that the cell wall will be formed, and thus callus has cells with different ploidy levels as well as cells with different kinds of chromosome. Where part of the chromosome have replicated twice, thrice, and part of the chromosome which hasn't replicated at all. So therefore, you're having different kinds of cells in the callus. And if you follow the usual method of plant regeneration, you can get different kinds of plants. If you apply somaclonal or you want to get variation, that's a great method. But so is somatic embryogenesis. But I'll come to that later. But first is that if you're going through direct somatic embryogenesis, whereas there is no intervening callus phase and the cell itself is getting transformed into a somatic embryo, the plant so produced will be mirror image of the mother plant. Now just imagine what kind of application it has in the field of micropropagation, where you want to clone plants. You have one plant, you'll appreciate that the orange variety, which is with very few seeds or no seeds, that has originated as a natural mutation. So there is just one plant with that trait, and you want millions of plants with that trait. How will you go about? Seed propagation is not going to give you identical plants. It's only that you have to go vegetatively. So if you can follow the method of somatic embryogenesis, you can produce large number of plants. Same hold true for vegetatively propagated plants. For example, banana is traditionally propagated by suckers. The plant produces almost 3 suckers in a year's time. The sucker weighs 2 to 3 kg. It is born in the soil and therefore it is exposed to soil-borne pathogens all the time. Now in India, the banana cultivation has gone many folds. Of course, we are not following somatic embryogenesis method for that. That is auxiliary method, but I'm talking of the future. Banana is susceptible to fusarium. Now, what is being tried, what is in research and something which is required is that can we treat these cells with mutagens and get mutants, some of which might be resistant to fusarium. And if you get one such mutant, and that is being tried in bioreactors through somatic embryogenesis because of the large number of plants that can be tested. So once you get one plant, you will perhaps find an answer to fusarium resistance in banana or many other diseases and many other plants. Now again, one thing which we are facing in micropropagation is transplantation losses. You need to have very good greenhouses. Your tissue culture facilities are at certain places because you also require trained manpower, you want to have sophisticated structure. So tissue culture labs can't be everywhere. So what we do is we acclimatize the plant, we take them out, we do the primary hardening in the place closer to the tissue culture lab, and thereafter we take them to the place where they are to be transplanted. Now, what is happening today that we are transferring truckload of tissue culture propagated plants in the fields where plants are to be propagated. we normally set up secondary hardening units over there, keep the plants there for a few days, and thereafter they are given to the farmer. But what one is aiming at in years to come, can we convert these propagules into seeds, into synthetic seeds? And this picture depicts that the small embryos, they are encapsulated into a gel-like structure which has all the nutrients for the early growth of the plant. Now, the overall success is restricted, and this is a researchable issue, that you are able to get seeds. Like then it will be, you want to produce plants in Delhi and you want to transplant them to Bihar, then it will be in 1 kg you are saying there are 10,000 or 20,000 somatic embryos converted into synthetic seeds. Your cost of transportation will come down, mortalities will come down, and that's how the technology will become more acceptable to the farmer. So this is a future technology. Apart from this, somatic embryogenesis has a lot of potential in other emerging technologies such as that of genetic engineering. There again, what you do is you transform a small tissue, and from the transformed tissue you get plants. You'll learn more about it, how the technology is carried out. It's not a simple technology, and therefore you have to, it's not that the transformation will occur where you require. Transformation can occur anywhere in the chromosome, and therefore you need large number of transformants. And those transformed plants, thereafter you need to multiply them in very large numbers to see their performance in the field. And that is again, somatic embryogenesis can play a very important role. Now, as in many fields of plant tissue culture, there are still many unanswered questions. I have said earlier that we have somatic embryogenesis protocol for over 500 species, but still there are many important species for which we don't have protocols. We need protocols and efficient protocols. For example, in case of date palm, starting from a single tissue, even after 3 years through somatic embryogenesis, you can't even produce 100 plants. Now that I won't call an efficient protocol. It's still a recalcitrant species and you need better protocols for it. In many cases, somatic embryogenesis, it is also prone to high chances of mutation, which you definitely don't want if your objective is cloning. That is very good if you're looking for variation. It's an efficient technology, but if your purpose is cloning, then you have to do things differently. It's also like theoretically, we say that cells can multiply indefinitely without loss of morphogenic potential. But in practice, we also find that in many species, with the passage of time, the callus cells, they loses ability to form embryos. So there again, it restricts the number of cells that you can actually get. Synchronization of embryo formation is still a problem, and many a times you get albino or abnormal shoots during regeneration of these embryos. So students, in this module you have learned how the somatic embryos are formed, how do you induce those as well as induce callusing followed by somatic embryogenesis, and thereafter you also induce synchronization of embryo formation. Somatic embryogenesis as a process has played a very important role in understanding differentiation as a whole. As for other tissue culture technologies, there are a lot of factors which affect somatic embryogenesis, right from your explant type, media, conditions in which you maintain your cultures, the way you keep your explant, and so on. The technology has been developed for many species, but to realize its full potential, we also have to move towards other disciplines, especially making synthetic seeds out of it. While the technology to some extent has been developed for synthetic seed formation, it still has many gaps before it can be commercialized. So once I'm sure this is commercialized, or you have commercial method of encapsulating somatic embryos, and when I say commercial methods are available, but then you can't store seeds like seeds. So you have to develop a combination, a gelling agent with nutrients, which stays for 6 months or so, and then you can use them as seeds. So once that is done, the application of somatic embryogenesis for plant propagation, for plant breeding is bound to increase. Somatic embryogenesis also has a very important role if you look at developing new kinds of plants and for genetic engineering. So thank you, students. I hope you have enjoyed this talk and are excited towards this exciting field of somatic embryogenesis.